Hello, my name is Michael Podvinec, and I'm heading the Biozentrum Research IT Technology platform at the University of Basel. Today, I would like to give you a brief introduction into the concept of the research data lifecycle. So, the idea of a research data lifecycle is something that originated around 2008. And this idea was originally brought up by the UK Data Service. The research data lifecycle consists of a 5 to 7 step model. There are some variations, and it is a very useful tool to think about the life of research data and also the different phases that data sets in research go through, and what kind of concerns you have at each of these stages. So from about 2015 on, this became really pretty prevalent when people started talking about data management. And you can see I just copied a number of research data lifecycle graphs from different universities, different societies, different organizations. And you can see it is a variation on the same theme. Sometimes it's a little bit more colorful. Sometimes there are more steps or less steps. Institut Pasteur, of course, has a version in French. Then there are some versions that are a little bit more complicated, sort of like an onion shell model of cycles within cycles. And I also found one that, well, it is a cycle, but they made it look pretty linear in some way. So anyhow, the research data lifecycle is a tool that helps you imagine the needs of research data, and it works across all disciplines. It's a really abstract concept, so it's being used a lot in Europe, in US, in the UK, also here in Switzerland. So the version of the research data lifecycle that I'm going to use in my talk is the one that we have here at the University of Basel. It's a 5-step model, so a little bit simplified, I would say. And I would like to just go through these steps and explain them a little bit and illustrate what the concerns at these different steps are. So when you start, well, the research data cycle, well, maybe it's important to say also the whole idea of a, I mean, I don't want to go into semantics, but the idea of calling this research data life cycle is interesting from the viewpoint that it kind of embodies the idea that science builds on top of the science that came before it. And just like that, not only knowledge gets reused, but ideally also data gets reused to create new research projects, to create new science. So the first phase, is the phase where you plan a new research project. Some other entities call this conceptualizing or receiving data. And this is really the phase where you think about a new research project, and you should think about what kind of data am I going to create and am I going to use in this research project? And also, how much data is it going to be? Is this existing data? Is this data that I can reuse, that I found somewhere that I'm allowed to use, or that we maybe have in the laboratory already? And typically, what kind of data on top of this will be generated, and with what kind of instruments, what kind of data modalities are these? How will the data be collected? How will it be organized and managed? So it's thinking about kind of the what and the how of data creation that is going to happen. And this is the right moment to do this is when you conceptualize a new research project, because depending on what you're doing, I mean, life science research has become extremely data rich in the last 15 to 20 years. So your scientific decisions may have quite an impact on the requirements, on the infrastructure you need to appropriately handle your data. So it's really never too early to think about these aspects as well. What you should think about as well are ethical, legal, and security concerns, if there are any, in terms of the types of data that you have. Is this data that is intellectual property, maybe of a company you're collaborating with, or is this personally identifying information or medical information from patients, then you need to have the adequate safeguards in place to handle this data. And these kind of thoughts and concerns, this is typically what funding agencies ask you nowadays to provide in what is called the data management plan. And that's really become part of planning a project to be funded. So the Swiss National Science Foundation, probably the most relevant funding agency for the people listening to this, have required data management plans since 2017. The format consists of 12 questions divided into 4 categories that essentially just follow exactly this. Describe the types of data, describe how you will work with them during the active research phase, how will you make them, how will you protect them, how will you make accessible what can be made accessible. So the data management plan belongs to a funded research project, but it is, as for now, it's not a part of the scientific evaluation. So if you ask for funds for a project, it will be judged on the scientific merits and not on the quality, on the strength of the DMP. However, a DMP is supposed to be there, and once it's supposed to be updated while you are doing this project, because I mean, through 4 or 5 years of a research project, a lot of things can and will change. And at the end of your research project, we've already seen now with the projects that started in 2017, the National Science Foundation actually does go back and looks into these DMPs, which then become final, and checks, did you actually do what you promised to do? Or they will point out to you what is kind of where there are disparities. There was a change quite recently in that you do not have to provide a data management plan any longer when you submit a research proposal, but you only have to provide it when the proposal has been approved, has been granted in principle. So this is good in the sense that it reduces unnecessarily writing a document for something that will not be funded, possibly. But at the same time, we discussed this in the research data network here at the University of Basel, and we're a little bit concerned. There is a certain danger that anything you need to think about data and its management is sort of pushed back and comes later after you've written your scientific proposal, after you've received funds. You kind of have to make do with what you already wrote there and the money you got to adequately manage your data. So I think even if you have to write the DMP later, thinking about data and how you want to manage it should still be included in the ideation phase for the research project. Also a quick note, the Swiss National Science Foundation allows to budget for 10,000 Swiss francs for data curation, data management and data curation efforts, mostly geared towards getting data ready to be published. The situation with the EU programs, Horizon 2020 and now Horizon Europe, are very similar. They started phasing in open research data. I call this the ORD pilot from 2014 to 2017. After 2017, it became mandatory to provide a DMP, and it still is mandatory. So even maybe a little bit more strict than things currently are in Switzerland. And if we look across the Atlantic, in the US, the NIH were probably one of the first agencies in 2003 to provide the first kind of requirement for what they call a data management strategy. And they have just renewed their rules and regulations for 2023. And they are quite a bit stricter now than what we have here. So for instance, this is like the first time where they're also saying if you're doing research, you have to provide a DMS. If you are providing a DMS, we are going to check into this during the regular reporting intervals, and we are going to check that you do what you promised to do. There are even provisions in there that NIH can terminate the agreement if there is really if the data management strategy is not considered at all. Let's move from the ideation phase to the active research phase. So where we create data, evaluate data, collaborate with people, and store data. This is really sort of the core of research, making experiments, making observations. A lot of the majority maybe of data will be generated in this phase. Data will undergo transformations. You bring data together, you convert it, you clean things up, you validate data sets, and all of that also kind of starts the creation of metadata. So data about data, describing your data sets in a way that you and that other people can figure out what is in them. The data gets analyzed, the data gets interpreted. So basically from samples, from raw measurements, insights are generated. And then results will be published eventually, or maybe there is a patent somewhere down the line. And during this time, data typically is stored in what we call active storage. So storage provided by your university IT department, for instance, or maybe by your local department, something like this, but not commonly in like public or in public repositories. And what we're talking about here is actually really nothing new, because documenting your research in an appropriate way, this is something that we've done since centuries as scientists. Here on the right-hand side, you can see this is a page of Sir Isaac Newton's lab notebook. By the way, a really fascinating resource and a good example as well for data reuse. It's astonishingly readable. So people have always written laboratory notebooks. Data was recorded maybe not as microscopy data, but as drawings or paintings, and calculations did not result in files and data sets, but maybe in books with tables like Tycho Brahe, who you can see down there, who observed the planets. So in a way, documenting your research is nothing new. It's not extremely trivial to do this well. I mean, the idea is really that you document your research so that somebody who is skilled in the art can understand what happened. So maybe your supervisor, or maybe a collaborator, or maybe somebody who comes after you and picks up the project. Or this could also be, this happened to me, you yourself, when you get an email from an old collaborator 7 years after you did something together, and they say, hey, great news, we finished our part of the work, and we'd like to submit the paper by the end of the month. Could you just, quickly summarize the stuff you did for us and write the materials and methods and kind of interpret the data a bit? And then you go back at this time really to paper lab books, and you have to sort of make sense of the things that back then you thought you documented really well. So it's not that easy to do good documentation. And the data that you generate, where do you store all this data? So there are different data management systems. In life science, we have sort of these 3 pillars, but many software systems in the end, they sort of stand on 2 or more of these pillars. But roughly conceptually, one thing is called electronic lab notebooks. So yeah, Isaac Newton wrote everything in paper. We're doing basically the same thing, but we're no longer writing in paper. We're writing things digitally. Everything has become digital in a way. So electronic lab notebooks, this is really where you put the narrative of your research, the who did what, when, why, what was the outcome, what was observed, what was what we expected, unexpected things, and what are the next steps that are we going to take. Also protocols, descriptions, and what I sometimes call like qualitative data. So maybe not microscopy pictures that you want to do some image analysis on, but just maybe something to remind you when you look at this data, oh, this is what the cells looked like at this stage. The second pillar is something that is called laboratory information management system. Here it's less about describing what you did and more describing about what you have. So this is kind of a repository of information about the materials in your lab, the samples in your lab. Things are important like provenance. This is the RNA that was isolated from the cells that came from the liver, from this experimental animal, this sort of thing. So keeping track of the workflows that happened. Finally, the third pillar is called the scientific data management system. And this is where a LIMS sort of has physical objects in its scope. An SDMS is maybe more about data objects. So storing and annotating large data sets, then typically also providing good search and good API functionality to access the data. So this is kind of the gold-plated solution. If you have these kind of systems for the type of data that you have, this is where you should put them. But in reality, that's not always the case. So part of your data will always be stored in files on some kind of file system. So maybe not in USB drive stacked like this, but our recommendation really is talk to your IT providers, work with them to use centrally managed storage where you have professional grade hardware and professionals behind that who manage these things adequately, who make sure that there is an adequate backup strategy, that data can be recovered, that data is highly available, rather than trying to roll something on your own in the lab. This is what we always recommend our groups. For this, you also need to understand and come quite early and talk with your IT providers, whether this is a research IT unit, whether this is IT services, whether this is scientific computing support. Try to get a grasp about what type of data you have, what size of data you have, because these data sets can be really large and they may require significant effort on the side of the infrastructure provider to be able to give you the storage you need. And then once you have these data, or once you have this storage, make sure it doesn't get too messy. So think ahead of time about a hierarchical and logical structure to organize your data. Also, ideally separating things that are still being worked on and things that have been completed, maybe even earmarking data sets that are making it into a particular publication. And also, the earlier you clean up things you don't need anymore. I mean, I'm not talking about deleting raw data. I'm talking about, I don't know, microscopy recordings you made that were completely wrongly illuminated, things like that, things that are just unusable. Really try to clean up as early as possible, because the longer these things stay on disk, the less sure you are in the end what this data set was, and if you really still need it, or if you can really delete it. And in doubt, I mean, we biologists, we're butterfly collectors, so we're going to keep everything. So version files using a file naming convention, or ideally, if appropriate, using something like Git. This is a much better solution, a much more flexible solution. And in general, use a naming convention for your file names. Ideally, one that all of your group or one that all of your collaborators use, so that some minimal information about what is this data set, where did it originate from, is already stored in the file name. Yes, let's move to phase 3. Conserve, annotate, curate, and archive the data. So we're slowly finishing up. We have data sets that will make it into a publication. We want to put data into public repositories. So it's about preserving data. It's about storing data. This is the time where you complete the annotation of your data with metadata. Some of this metadata will only be, you will only be to annotate this after you've done a certain amount of analysis on your data. Select the data that has long-term value. Think about file formats. Don't use super proprietary formats, but rather use open formats as much as you can. And in this way, you're getting your data ready to be deposited either locally in an archive, if this is data you cannot or don't want to share, or if this is data that should make it into a public repository. One small comment about metadata. This is an example I always like. This is a picture from Wikipedia. And if you just look at this picture, you're seeing certain things. You can maybe make some educated guesses about things, but you don't really know that much. It's a black and white picture, which may tell you a little bit about the period, also how the people are dressed. This may tell you a bit about their socioeconomic status. If you look a bit more carefully, you may see an American flag in the background and even the seal of the President of the United States. But apart from that, this is just a black and white picture. But since also Wikipedia has metadata, if we would scroll down here, what you would get is the metadata that was annotated with this file. And this is technical data, sort of what file format is this in, what resolution and versions are there of this file, but also information about the content and the origin and the creator and the license. So the content will tell you that this is actually Lyndon B. Johnson in 1964 signing the Civil Rights Act, and Martin Luther King is standing behind him, watching over his shoulder. And the image was taken by the press office of the of the White House, and like all things that have been produced by the US government, this can be used and shared without any restrictions. So also the reuse license is in here. There could be more metadata about this. And this is clearly not a life science example. But if you want to find out about metadata standards, there are 2 catalogs that can be recommended. But also, it's not that easy a business because there are many metadata standards. So if you look in the RD Alliance, you will find there are 15 entries sorted under life sciences. And if you start opening these up, like the MIBBI is actually an agglomeration of 39 different metadata standards. So some of these standards are a little bit arcane. Some are very well known, like Miami standard for microarrays, which is already 20 years old. MZXML and colleagues for proteomics data also, yeah, probably getting towards 20 years old. PDB, the maybe first well-defined file format in structural biology, or OME XML, or DICOM for medical images. So there are well-defined standards, but there is just a huge, overwhelming wealth of standards. It's not that easy to pick, for sure. XKCD, like almost always, has the answer for this. But I think the good news hidden in this is what we're seeing over and over as fields mature. There are really metadata standards that are being used by the community and that then establish themselves as the de facto standard. All right, moving to publishing and sharing your data. So your data is well annotated. It may be accompanies a publication and you want to put it into a public repository. So the question here is usually, how do you select the correct repository, the best repository for your data to fulfill the fair data requirement? And this slide here shows maybe sort of 2 aspects of the 2 sides of the same coin. Conceptually, what you should do, well, the repository should support fair data. This is something that publishers are asking for. This is something that funding agencies are asking for. And it's also something that makes sense. And when you select your repository, the recommendation is to choose a domain-specific repository rather than sort of a general purpose, we take all kind of data sets repository. To also choose a common repository, something that is operated by some common entity, rather than putting things into the data repository of your institution, if your institution has one, and to see what other people in your community are using as repositories. So basically, these 3 points all amount to the same thing. Put your data in the place where it is easiest to be found again by other people, to be reused, and don't sort of hide it like somewhere in the in the cupboard in the basement of your institute, right? Figuratively speaking. The Swiss National Science Foundation has very clear guidelines what they consider a valid repository. This is published on their website. So this is also something to take into account when you want to make sure you don't run into issues or discussions with the SNSF later. And to help you find the right repository, there is something like re3data.org. This is the Register of Research Data Repositories, which I would highly recommend to go there and see what repositories are around to be able to take up your data. What a lot of people use, and it's probably not the best solution, but it's by far the worst, it's by far not the worst solution, is putting your data in Zenodo. This is kind of the general purpose repository everybody knows and loves because it's just so easy to deposit. It's operated by CERN, so it's publicly accessible. You can put any data in there, no questions asked. This is great. The downside is, since it is so general purpose, the metadata that you're providing to go with your data mostly has to be provided in terms of like a text file. So there's very little ways of structuring metadata in relations to other data sets. Something I would just like to popularize here a little bit is that the EBI has recently launched a general purpose repository, but for the life sciences. So it's called EBI Biostudies, which they position as this is where you put data you cannot put in any of the other EBI repositories. And I would really suggest you have a look at this and maybe consider using this instead of Zenodo for data that is biomedical data, because it allows much better integration with other databases. And it is in fact recommended by Elixir as a deposition database. And now we're closing the circle. So once the data has been published, it is there to be discovered, to be reused. And also I think, so the data can be used if you want to validate a new method or do some pilot experiments. But I think also something that we maybe don't think enough about is you can also use these data sets for training and for teaching as good example data sets. And what is still quite important, when you do this, always respect the usage license and only do with this data what you're allowed to do. So summing up, I think the research data lifecycle is a very useful tool to conceptualize what must happen with research data in order to get it into a state where it becomes easily reusable. And there are some important quality checkpoints there. The first one, obviously, is when you start out on a project, you need to think about the data management plan because it's required by funding agencies, but also because it is a good process to become aware about where some challenges maybe in the management, in the annotation of your data may lie. And then you happily do your research, and then at some point you get to the point where your data needs to be published. So under the SNSF rules, for instance, this is latest when a scientific publication is made that was created with the use of this data, and SNSF asks that you publish all the data that is underlying this paper. So roughly said, anything that goes into a figure, but of course not data that for some reason is protected and cannot be published. And SNSF asks that you document the data sets in a way that is adequate, that others can understand and reuse it. So that's a bit of a difficult definition, but at least in spirit, this is exactly what we want. What this means is if you're doing all this work, then at the time of publication or at the time of the end of your project, this is substantial effort. The data curation effort is real. This is time. This is effort to complete the metadata annotation, to bring the data sets together, to get a clear overview, what is what needs to go where, maybe transform some data sets into other file formats. This is a lot of work. And this can be really complicated because if your project has been running for 4 to 5 years, maybe also the researchers that originally did the work are no longer in your group. So somebody kind of has to pick up and understand what other people did. So what I would like to popularize is the best way to handle this is not to do it here at the very end, but instead understand that annotating and organizing your data is an activity that you do regularly as you're doing your research in the active research phase, done by the experimenter who knows best what exactly this data set is, providing metadata about the experiment, flagging data that are making it into a paper, which makes deposition easier later. And you can do this, of course, according to the context that the DMP for the project provides us. Or what I would like to suggest, what I think would be a good idea, is to work towards having a kind of data management, default data management plan that you use in your research group. And so that the processes are clear, they're always the same. And from such a DMP, you can very easily create a project data management plan that the funding agencies will ask from you. So I think long-term, this is the best way to go to make sure you're not ending up sort of almost at the end of the loop and have to figure out a lot of things with a lot of data sets that already are a couple of years old. So yes, in the end, I guess the question remains, so does data curation spark joy? Like Marie Kondo says, who made a career out of cleaning up and arranging people's apartments. And of course, data management, some people say it's a boring topic. And well, yeah, it's work, it needs to be done. It's maybe not always rewarding, but it's the same thing like if you never clean up your kitchen, it's going to be harder and harder to cook a really delicious meal. So cleaning up the kitchen is just part of the creating delicious food process. And I don't like emptying the dishwasher, but I understand. Well, actually, I'm happy we have a dishwasher, and I understand that this is the prerequisite to cooking something really nice. So I think we have to look at data curation, data management, data annotation as just an integral part of doing science. Okay, thank you very much.